Thanks a lot, Arnie. Thanks for inviting me to speak here. And Arnie also told me that I don't have to tell everyone to ask questions because you already know that that's the format. So I'm going to tell you about E. coli and bacterial growth and how bacteria survive in changing environments. And maybe just to start, give you kind of an overview of some of the questions, because this conference is about the questions and the problems that physicists can contribute to. So I hope that as we go along, you'll see some big open areas where a lot of contribution can come. So broadly speaking, these are the kind of big topics I'm interested in, and I think today's talk will revolve around. So one big set of topics is survival strategies and molecular mechanisms. So the question being, how do organisms survive? What kind of molecular mechanisms do organisms have that enable survival? The specific question in microorganisms is you have a fluctuating environment. Conditions external to the organism are changing with certain statistical properties, and we'd like to know how does the organism deal with that. Practical question is, how predictive can one be about organismal growth in fluctuating conditions? So one basic question is, what are the basic ingredients that you need in order to make predictions about growth? And as you'll see, it's a more complicated problem than you might think initially. And I think the longest term question here is, how do mechanisms actually evolve? So I'll introduce a few specific molecular mechanisms, but we really don't understand how any of these mechanisms have evolved. We can build phylogenetic trees for proteins and see relations, but I really don't know of too many great examples where we understand how something so beautifully tuned or well-developed molecularly has actually evolved stepwise. I think that's a huge problem. That's definitely open. So, okay. I have a couple of parts of this talk. So the first one is on memory in bacterial adaptations. And this is really two interrelated projects. The experiment side of this is by Guillaume Lambert, and that's on the observation of memory in the sugar operons in bacteria. And the other half of this is theory, which was done with Antun Skenada, who's actually right there. And that's on evolutionary phase transitions. So I will tell you about both of these. And in some sense, the experiments really inspire the theory in this case. So I'm going to start by just talking broadly about how does a microorganism survive when the external environment changes. And the cellular state may not be adapted for some stress, which means that certain proteins, certain genes have to be turned on when the environment changes. And the question is, how does the organism go about doing that? And there's different classes of mechanisms. So one big class we call responsive switches. This is when the organism knows that there has a sensor, it receives a signal and sees that the environment changed, and it knows what to do about that change. It knows it needs to turn on a specific gene or pathway to do that. So there's a response, there's a sensing of the environment, and there's a coupled genetic pathway that's going to enable adaptation. That's responsive. On the other extreme is a purely stochastic strategy, where the organism doesn't use any sensors and just randomly turns genes on or off. Now that may seem not like a very good strategy at all, and it wouldn't be a very good strategy if it were just a single organism doing it. But if it's an entire population of 1 billion organisms that are doing that, and they can all switch on different pathways, which are good in different conditions, then what you have there is a population that's maintaining diversity and allowing you to actually survive to very broad range of environments. And when the environment does change, this process will select for subpopulations that have turned on the right genes. Okay, so is that clear? That's kind of like a totally different strategy. It's not triggered by the environment. So there's no sensing here. And the actual molecular mechanism that implements the strategy could be very different. So there could be genetic implementations of the strategy. There could be epigenetic ones as well. Okay, so the point here is really about the strategy. It's the class of the strategy rather than the mechanism. So in reality, biology takes advantage of all possible degrees of freedom that it can, so as far as evolution goes. So you can actually have cases where you have mixtures of these strategies, and those will adapt in certain ways. And so one big question is, when is it advantageous to use one or the other? When is it good to use mixtures? And many questions about all that. Okay, so I'm going to, actually the first part of this talk will mainly be on responsive switching. The second part, I'll introduce a stochastic switch, and that'll be kind of the overall organization. So I'll introduce the best studied responsive switch in bacteria, which is the lac operon. Has that been discussed already? No. Okay. So this is basically the first gene regulatory circuit that was fully elucidated. And the experiments I'm going to show you in a second actually led to the discovery of gene regulation. And now we know how this works. So this is a huge number of papers. So basically, usually when E. coli is growing in a medium that contains glucose, it can actually build everything that it needs, all the carbon molecules that it needs directly from glucose. So you can simply feed it glucose and it will do everything, which is quite amazing already. We're not actually going to discuss that. What we're going to discuss is what happens when you give it a different carbon source. Okay, so if you give it a different carbon, a different sugar, E. coli will have to turn on specific genes that enable it to metabolize that sugar. So lactose is one of the sugars that was initially studied by Jacques Monod, and what he and others found was that in fact there are eventually found 3 genes that together enable adaptation to lactose. And those genes are now called, that's why it's unintuitive, yes, this particular one. So LAC-Z is the beta-galactosidase, and that is an enzyme that cleaves lactose into glucose and galactose, and then those 2 sugars can be metabolized. And LAC-Y is a permease, and that's a protein that goes to the membrane and then brings in more sugar. Okay, and it specifically binds beta-galactosides. So it binds lactose and a few other analogs. Okay, and this operon is normally turned off. So there's a repressor protein that sits right here and makes sure that the cell does not produce any of this, because apparently it has some costs, and those costs have been measured. So there's actually a cost to producing these enzymes and to making a lot of them. So the cell keeps that operon turned off and only turns it on when it needs to. The way it gets turned on is that if there's some lactose in the environment, lactose can get into the cell in some non-specific pathways that bring lactose in. And then what happens is there's always a basal amount of LAC-Z in the cell, basal amount of beta-galactosidase. And what LAC-Z will do to that few molecules of lactose that come in is cleave them, but it'll also make another side path. And there are a couple of reactions that it catalyzes, and it also catalyzes conversion to something called allolactose, which is a slight modification of lactose. And allolactose binds this LAC-I repressor, which is at that point sitting here, and it derepresses at that point. So the LAC-I falls off, and some of the transcript of this operon is made, and some more LAC-Z is made, and then you start making the permease. And the permease goes to the membrane, binds more lactose, and so you start concentrating lactose in the cell, making more allolactose as well as glucose, and then more time is spent in the derepressed state. Okay, and then more permease is made. So you see there's this very strong positive feedback loop here that turns on this operon and then enables growth on lactose. Okay, So that's the system. Let me show you some of the experiments, initial experiments from Jacques Monod in the 40s. These were published as part of his PhD thesis. So, and this is, which is great. People actually still refer to Monod's thesis when this is a review that he wrote a few years later. And what he was doing is, and this is before they understood how this all works. He would grow E. coli in certain conditions, for example, in pure glucose, or in pure arabinose, or in pure any sugar. And then he would shift the conditions abruptly. And what he found was that in this particular experiment, he's shifting from growth in arabinose, and he would shift, in this case, to growth in glucose. And that's this curve here. And this is a log scale, and this is time. So growth on a log scale that's linear is going to be exponential growth. And so you see that you have exponential growth in glucose from the outset. So what he would find is whenever you switch to glucose, he wouldn't see any delays, and you just have immediate growth in glucose. It's exponential. If you switched between different sugars such as arabinose and in this case xylose, sugars that are more complex than glucose, he found that there was a lag and there kind of a gradual acceleration of growth eventually to steady state exponential growth. And these lag times depended on which sugars you switched among, and they were all typically 1 to 2 hour lag times. So this is for pure switches between two carbon sources. He also discovered another phenomenon, which is if you grow in mixtures, you actually give both sugars to begin with. You give both glucose and another carbon source. Okay, so here's a mixture. Then what he found was that there was first one phase of growth, then a plateau, and then another phase of growth. And the more glucose he gave, the longer the first phase of growth. Okay, so here's a lot of glucose, a lot of growth before the plateau. And so the interpretation here is that bacteria preferentially consume glucose. It's the first thing they want to consume. And of course, he measured the glucose concentration and saw that indeed glucose is being consumed first before you go through this lag. So he called this dioxic growth. Okay, so that's the basic observation. Yes? So in this case, he's controlling the mixture, the actual ratio of the two. Yeah. Okay, so the typical, these plateaus here are comparable to these as far as time, and they're about 1 to 2 hours. So eventually this led to the discovery of gene regulation, the fact that you're turning on specific genes at certain times. Okay, we're not going to go through, of course, all that. But gene regulation itself, as you can see, is obviously useful for turning things on. But in reality, we know that lac operon is repressed, which means that at some point it also gets turned off. And that means that really, you need gene regulation in order to grow in a changing environment. And so most of the work that had been done on gene regulation has really come from single shifts of 1 or 2 conditions. And so we haven't yet learned how organisms actually adapt to continuous fluctuations in time. And so one of the things we wanted to study was how that happens in E. coli. And to make continuous fluctuations in time, it becomes very useful to have a device that allows you to do that, rather than having to spin the cells down at every point, perturbing them, resuspending them in a new media. Okay, so you want to kind of do this seamlessly. And so Guillaume was working to develop a device to do that, and he took as his basic design something that was called a mother machine, which was invented a few years before by Seok-Jun Jun. This was a device that you may have, I don't know if when you were talking about aging, but this really was invented to study aging in E. coli. And it was a device in which you had single cell chambers that fit exactly one bacteria, and you could watch them pushing out and dividing and tracking only this remaining cell here, which is called the mother cell, and removing all the daughter cells. So this device was invented to study aging, because you could ask how many cell divisions can this mother cell undergo before it stops dividing, or does it get slower and slower and slower as it grows? And it turns out that this mother cell does not get slower. It keeps dividing. You can have it maintained for tens to hundreds of generations without any problems. So there's a flow in this device that
What are the problems that after generation?
the problem is that there's a rate of filamentation that occurs in bacteria, at least in these chambers and pretty much in all the PDMS-based experiments. And once a cell filaments in this chamber, you'll actually see one of those events a little bit later. It creates a very long cell body, and eventually, so it grows to much bigger size than the actual chamber, starts sticking out here, and the flow pushes that out.
So if you make a cozier and cozier chamber, can it live forever then?
Yeah, the thinking is that either there could be a spontaneous rate of filamentation, which then you could take as some kind of aging, but in fact it's not. It's a process that seems to be able to occur at any time, so it's not real aging. Although there's.
The system breaks you eventually.
Yeah, that's the limitation at the moment. So.
Wasn't there a time that people thought there were mother clones and daughter clones? That even though it was binary fission,
Yeah.
one of those two was destined to peter out in its lineage, while and that was sort of looked at under the microscope without the flow. Like
Yeah.
other machines,
So what I'm
filters, yeah.
right. Well, so in bacteria, what I'm familiar with is this work of Francois Taddet that kind of tracked poles.
Yeah.
So saying there's an old pole and a new pole.
Right.
So when a cell divides, E. coli cell divides in the middle, there are two new poles formed in the middle. And if you track those, you can kind of assign age to something that's symmetric. That was the idea. So Sakjun actually, his device was meant to kind of test that for a longer period of time than you could do in agar pads. That was the idea. So we wanted to actually, and you'll see in the second part of this, we want to study a system in which you actually have a small population, because eventually we also want to study stochastic switches, and you need a population in order to be able to leverage many cells. So a mother machine is not really an ideal device for us because it's really just treating cells in isolation. So we want to have some small population. So Guillaume modified this to have larger sizes here, and we still want to keep monolayers so you could track all the cells. And then he basically designed this so that you can rapidly change the nutrients that are in this flow, and so you can very rapidly change the environment. So the basic idea here, so this is just to show you that,
So when you see the flow outside, when you say the environment change, like the bacteria have to censor that, right? So how do they censor what's happening in the immediate environment? Because it's
in the flow.
Yeah.
Right. So there's diffusion in this case, and we're flowing in a small molecule so lactose is a small molecule, and it has a rapid diffusion time in these chambers. So I'll show you that right now, in fact. So what we did is, in order to measure that exact process, flowed in a fluorescent molecule that's roughly the same size as lactose. And you'll see here, chambers are filled with this molecule. And then we're going to just change the medium so that the medium no longer contains that molecule, while again flowing at the same rapid rate through this chamber. And so what you'll see is, I'm running a movie here, turning the flow off, and then you see that fluorescence vanishes because now you had diffusion out. Okay, and then you measure the fluorescence rate, you measure the fluorescence intensity here, and you see that within about 10 seconds, you've turned over this chamber. So the idea is that all the cells in that chamber would now experience a different nutrient environment. Okay, so if you can, once you can do that, so you can grow out the cells, and I'll just show you growth, just so you can visually see what happens when we just remove any carbon source from the cells. So first we're growing them in glucose, and they're dividing happily for 3 hours. Now we remove glucose, and they stop, and then we give them glucose again, and they grow. So that's 12 hours right there. Okay, so basically you can just randomly expose them to these changes, and in some cases they'll react. Okay, so, oops, yeah. Okay, so now in order to, what we want to kind of do is run a Monod type of experiment, but keep going back and forth. So switch from one sugar to another sugar and then back. and do that at different frequencies. And so I'm going to show you a plot of this kind here on the right, which is basically the quantification of the kind of movie you just saw. And so, the most dramatic thing you saw is this kind of flow of cells pushing. And so what we do is we quantify the amount of pushing that's happening at a certain cross-section here. And that's a cell speed. And that's proportional to the elongation rate of everything behind that point. Okay, so it's kind of a bulk measure of growth within chambers. So you see that they're pushing in glucose. This is an overnight growth in glucose, and this is the last 45 minutes of that. So then we switch them to lactose, and you see they stop. They go to 0. And then about 30 minutes later, you start seeing recovery. And within about 50 minutes, the cells are happily growing in lactose. Okay, so that's just the Monod kind of experiment where you make the switch. So this is you see a lag phase, recovery, and then full growth. So, yeah.
Is an experiment in which is demonstrate that you can switch from lactose to glucose again for the same bacteria, and if there is the plateau maintained?
Say it again?
No, I'm wondering if you switch from lactose and glucose for the same bacteria, there is a plateau gain, or there is some...
Oh, from lactose back to glucose. So in Monod's experiments, you couldn't see anything. Basically, if you switch from lactose to glucose, you would see purely exponential growth in glucose. That would be kind of the classic experiments. So that's like in his thesis. So if you repeat that experiment in microfluidics, that's this little inset here. So overnight growth in lactose, you're growing steadily. When you switch to glucose, you see a down blip for about a few minutes and then recovery. Okay, but it's not at all a delay, a growth lag. So it's probably associated with the fact that carbon fluxes have to shift in the cell.
This is learn something probably, right? There's a sort of learning.
Not at this stage. At this stage, I think it's purely carbon fluxes that are changing, most likely. But we don't exactly know. Yeah. Okay, so if you're going from glucose back to lactose, you go through these lags. And so now the question is, what happens if you do this again? And so we just did a fluctuation experiment where you grow for 4 hours every time. So overnight growth in glucose, you go to lactose, you recover, you have 4 hours of growth now in lactose, then you go to glucose for 4 hours, then you go back to lactose, and this time you don't have any lag. And then you go to glucose again, and lactose again, and glucose again. You never see lags again in this experiment. Okay, so the lag goes away, which suggests that the cells remember somehow that they saw lactose back here, even though they've been in glucose for 4 hours. And so the key time scale to know is that in this experiment, the cells are dividing about once an hour because they're growing in a minimal medium. They can actually grow faster than once an hour, but if they have to make everything out of carbon, out of pure glucose, then it takes them an hour to grow, to make one cell division. Yeah.
When you say that they remember, you mean that perhaps the enzymes which are processing these molecules are still there?
Yeah, that's right.
Or
And that's
because the bacteria somehow thinks that has to produce them even if it doesn't
Mhm.
kill the lactose.
Right. So the question is whether it's still actively producing them or just passively maintaining them. And you'll see the answer is passive in this case. But that's exactly right. So they clearly still have the capability to metabolize lactose. Yeah.
Maybe a bit of a silly question, but we have experiments, so there's a lag, a change over time, which we could store some glucose in our stomach. How much do bacteria have this ability to sort of reservoir carbon?
Yeah, that's a great question. And so you can do a calculation. I mean, so molecularly, there's no known mechanism for storage of glucose or lactose in E. coli. But you can also do a calculation to see, how quickly, if the cell is just packed with lactose, right, in steady state, how quickly does it consume all that lactose, right? And it'll take only a few seconds, turns out. So if you don't bring any more lactose in, you should cease growth very, very, very rapidly. Yeah. And that's the reason we can do that calculation is because people study the biochemistry of these enzymes really, really completely. So we actually know rate constants for every single reaction here. Yeah. Okay, so yeah, so now the question is where does this, how long does this memory last? So clearly, if you grow the cells out for very, very long times in glucose, you will eventually go back through lags when you see lactose again. So we repeated that experiment by increasing the time since the last lactose exposure. I showed this data point, which was 4 hours. That's where you had perfect, absolutely no lags. And so this is the growth delay. How long is the lag phase? And you see that if you go up to 4 hours, there's no lag phase. If you go to 6 or 8 or 10 hours, you start getting lags, and then you start getting recovery phases. And it takes you you need to not have seen lactose for about 12 hours before you start seeing the full lag phase, which was around 50 minutes. Okay, so you see that the memory lasts a certain amount of time. It's definitely multi-generational. Okay, so basically here it means that the cell is remembering the state of the great-grandmother cell, right? Remembers that it saw lactose, and the progeny 4 generations later never have to go through a lag. That's the basic idea. Now, the molecular side of this is that it's quite a simple mechanism. The lac proteins are extremely stable, so they have very low degradation rates, which means how often are they turned over in the cell. This was measured in the 60s or 50s, I think. So degradation times of about a week. That's quite amazing. This protein just remains completely stable. And what that means is when the cell divides, the daughter cells inherit half of that protein unless they're still producing it, right? And then those inherit half, so you have half and half and half. And it turns out that if you divide 4 times, so right now you have only a fraction, 1/16 of the amount that you had initially, that is sufficient to avoid lags. And that means that at that point the cell then starts making much, obviously turns on the lac operon and makes a lot of lac. So you can see this in different ways. You can overexpress lac-Z. This plus means overexpress, means you give extra copies of lac-Z to the cell that are constitutive, so they're not regulated. And you see, of course, that in that case, the cells absolutely don't have any lags when you switch. You can overexpress the permease, and you see that then you have shorter lags. Okay, so these molecular components determine whether or not you have memory. And you can also track fluorescently labeled permease. So you can track in single cells and see dilution of the permease and then turning on of the operon. And you can see that if you fluctuate rapidly, you don't fully dilute the permease. Okay, so you basically still maintain permease levels, and that allows you the capacity to avoid lags. So this is the kind of the molecular side.
I presume that the degradation of the beta-galactosidase is a regulated process in the sense that it could have been that it made an enzyme that was degraded rapidly, or it could have been that it made an enzyme that was stable. And so the stable enzymes would be selected for, and that suggests that this bacteria is in fact anticipating variations in the environment and makes a stable enzyme as a response to that variation.
Yeah.
So that it doesn't have to keep
That's right.
turning on and off and have lag.
That's right. That's right. That's the way we're that's basically the kind of hypothesis we're trying to test. Yeah, and to basically see whether that's, why is, so yeah, I think this will brings me.
Even the degradation of an enzyme is selectable.
Is selectable, right.
And is beneficial if it was stable.
Yes.
In a changing environment. So there's that kind of interesting implication that the bacteria is living in a changing environment because it experienced it.
Yeah. So what we'd like to, so just to follow up on that, basically we could ask, in what environments is it useful to have this kind of memory? When do you want to degrade the enzyme rapidly, and when do you want to maintain it for long periods of time? So that's going to be the subject of the second theoretical half.
Is there any actual, the wild type environment that we call that? Promiscuous bacteria, maybe that's
Yeah.
the point that it's somewhere everywhere, and that, so it has to deal with that. But
Yeah, I mean,
It has 2 environments. One is it lives in your gut, and the other is it lives in the sewer. Right?
And it has to transition.
It has 2 environments.
Transitions between these. Yeah.
Which one is which?
Yeah, so we don't really have people are studying the gut environment, and there's measurements of nutrients, and people know, for example, orders of certain sugars that occur in different stages in the gut. But
The other thing
yeah.
that's very different about those environments is the gut is anaerobic and the sewer is aerobic.
mm-hmm, mm-hmm.
So
Right.
I don't know if you ever did an anaerobic.
We have not done a purely anaerobic. No, we have not done anaerobic. I think that's interesting. Yeah. Yeah.
Are there any counterexamples to basically what Arnie said? Like, are there some proteins that are unstable, even though that they're constantly used or used fairly frequently in the environment, and aren't able to be passed on to next generations? Like, what makes some
Yeah,
proteins
yeah.
selected for and others not?
Yeah, there are examples of unstable proteins in E. coli, but we don't yet know enough about all the pathways. For example, if you just look at sugar, we don't understand yet all the stabilities of those proteins relative to that pathway. But for example, there's certain stress operons that will have a regulator that actually gets turned over very rapidly in order to enable, turning off this response. So I can give you some specific examples, but I can't say that there's enough knowledge of degradation in bacteria. Now in yeast, for example, there's, I think, better understanding of which pathways are degraded and aren't. So, but of course that's more complicated because there's whole ways to regulate that. So it's an interesting question, but we don't yet know. Yeah.
I think as a generalization, proteins that are folded have high peptide bonds from proteases that are specific. So most proteases have specificity. Trypsin is a lysine, and chymotrypsin is a hydrophobic amino acid. And they hide, if you have structure, it's very hard to get at the spots that you want. And if they are less folded or have unfolded regions, they then get degraded.
Mm-hmm.
So
Mm-hmm.
structure is one way to get at the thing you're talking about as a generalization. I don't know about E. coli or others.
Absolutely.
There's not an organelle like there is in eukaryotic cells where you degrade at low pH. Low pH is a great example of opening up structure. Right? That's why we go to low pH. And then the proteases just degrade it very rapidly. But the implication is protein structure, which we heard about yesterday, is in fact a protective mechanism for half-life.
Mm-hmm.
I don't know what
Yeah.
beta-gal looks like.
So
actually.
beta-gal is massive, and it's a tetramer. So that's another aspect of it, is that it's also making very stable protein-protein contacts into a larger tertiary, quaternary structure. So yeah.
Kind of in a way, it can't fully select for any type of protein. There's only specific proteins that you can selection for that can be passed down. Is that the idea maybe?
We think it's quite general, because if you just stabilize any protein, you could have this mechanism happen. So I think it's quite general. And the follow-up would be to understand globally which proteins do this and which aren't, and whether these larger structures contribute to that stability. Yeah.
There's an excellent correlation between the thermal stability of a protein. So take a purified protein, you raise the temperature, and it denatures. Right? The thermal stability of a protein is proportional to its half-life. Directly proportional. So if proteins that melt at 40 degrees are really going to be degraded and have short half-life. Proteins that melt at very high temperatures are going to be very stable in the cell.
Yep. Another question?
The environment fluctuates fast enough is that no response.
Yeah, so when you go to beyond the generation time and the fluctuations, there's a different kind of memory that actually acts, which enables you to turn on the protein despite the fluctuations. That's a separate thing that Guillaume found in these experiments that I'm not going to go into. But that's, I think, very interesting, and something we understand less than we do this. Basically, how do you manage to turn something on when the environment fluctuates? Why don't you saturate at an intermediate level that looks like a mixture? It turns out that you'll just induce. And in fact, when you do that, you'll see that lag times can become extremely short as well. So there's some very interesting things that happen at fast fluctuations that we don't understand. Okay, so I'm going to stick with this kind of intergenerational memory. And just say that, this is actually, we tested this in a few other sugars, and you see variants of the same theme here, that you see, eventually you see that the cells totally remember the past. Okay? So we're going to just focus on lac operon at this point, but just as a kind of model. So, and this gets to what Arnie said before, is that we have an emerging hypothesis here, is that gene expression levels may be tuned not only for the immediate conditions, but also provide memory for future generations. So one big question in the field has been what sets the gene expression levels of different genes? Why do you make that much of that protein? Why do you make that much of this other protein? Usually it's been understood in a cost-benefit analysis in a single environment. So you basically see how do the cells grow in glucose, how do they grow in lactose, what costs and benefits does gene expression have on a protein-by-protein basis, okay? And you balance those costs and see whether you're in some optimal expression level. In this case, what we're saying is that there's actually a second process here, which is the inheritance. If you make a lot of this protein, you can pass more of it to the daughter cells, and therefore the daughter cells will avoid lags. So in a fluctuating environment, avoiding lag is actually quite a good thing, because if I'm in a lag and you're growing, then you're exponentially outcompeting me, okay? So basically lags seem to be, there should be a very strong selection against lags if you can avoid them. That's the basic idea. So So we'd like to perform a cost-benefit analysis, but in a fluctuating condition that takes into account memory. And we'd like to know when does phenotypic memory of the kind that I showed you give you an advantage to cells in a fluctuating environment. So I'm going to turn to this theory now and write down what we think is the simplest model for sensing with memory. So kind of the simplest model that captures some of the things that I showed you. So what we need is two environments, a growth environment and a stress environment. And these will be, this stress in the case of the lac operon is, of course, the fact that you have to grow in lactose and you don't have the enzyme yet. So at that point, it's a stress. So growth, stress, and some fluctuating condition and time on this axis. And now we have to tell you what the cells are doing. So when they're stressed, the cells will turn on a stress response that enables them to grow. So if they don't have the stress response, it's open, this is off, the gene or network is off, and then in the stress, you turn it on with some rate, K on, and once you're on, you can grow with a rate B. So you can think B is the benefit for having the stress response on. The off cells cannot grow. Okay, so no arrow here. So that's the model. This is just, you could translate this into two differential equations, right, with no problems. So that's the model. In the growth, the cells degrade the stress response. So any cells that are on in the growth condition, the assumption is they don't need the stress response, so degrade it with a certain rate, K deg. And once you've degraded it, you can grow faster than you could grow when you were on. So there's a cost to being on when you don't need the stress response. There's a cost to producing all the proteins or having all this extra protein that you don't need, and that cost is captured with this C. When you degrade it, you now grow faster. Okay, so off cells grow with rate G, on cells have a growth cost. Okay, any questions? Yeah.
So what's the relationship between B and G?
Between B and G, you could take them to.
Which one do you expect is larger?
You could take, well, if you look at the lactose glucose.
Sorry, B compared to G minus 6, sorry.
Oh, this difference? Yeah, so, that really depends on the cost of these proteins. So, you can measure that in different cases. So, in the lac case, it could be a few percent. This cost could be a few percent, maybe up to 10%, but it depends on some details.
Yeah, but do you expect that in a stress condition, so the on cells will grow faster or smaller than, in the growth condition?
So if you're comparing G and B are usually quite comparable. As you saw, they grow happily in lactose once they adapt. They grow happily in glucose once they adapt.
Okay.
So those are the, yeah. So as far as this model, these are now parameters that we'd like to explore to understand the regimes of the model. We'd like to understand when this model has certain behaviors. That's the basic idea. Okay, so how do we analyze this model? So I want to give you first a really intuitive cost-benefit analysis, and then we'll look a little more in detail. So here's the network in case you need to remember it right up here. So how do you figure out how much memory is good? Well, first we consider the extremes. So consider a memoryless response. It's a response that totally doesn't remember the fact that it was on once the environment changes. So what does that mean? In the growth phase, we keep the response off. In the stress phase, we turn it on. And then once it's on, if you switch back to growth, you immediately degrade it. Okay, so KDAG would be very high, and then it's off, and then it gets turned on again. So notice every single time, if you have memoryless response, every single time you go through these lags, right? Okay, so that's the yellow area there is basically the cost of the memoryless response. Why? Because if I had memory, I would beat you during the yellow period. I'd already be on. So the cost of this lag phase is the benefit that I would have had if I were growing times the duration of my turning on rate here. So the cost of this lag phase is B times tau on. Okay, that's a simple hand-waving calculation. Memoryless response. Now we do a response with long-lived memory. So now you turn on, yeah.
The term from C times K deg is assuming that's very small. There's also a small.
You're right. You mean the fact that you're slightly degrading at all times, but you're producing it? Right. So, yeah, but the point is that really here, you degrade very, very rapidly, and then here you're turning on. Okay, so that any correction would be tiny in that case. Yeah. So again, hand waving calculation. Okay, so response with long-lived memory means we turn on the gene, maintain it indefinitely. Okay, so as much memory as you'd like. So that is actually fine because you avoid lags, but every period you're actually going through an entire environment in which you have turned on the stress response when you don't need it, right? So the cost in this case is the cost of memory, is the cost of the stress response when you don't need it, which was C, times the duration of the environment. Okay, so for the first time, the environment statistics have entered the calculation here, right? So this is something from the external environment, how quickly it fluctuates. So now, in these two limits, you can compare and see which one's bigger. So, in fact, I should stand over here. Okay, so in fact, memory is beneficial when the cost of memory, which is here, is less than the cost of lags. Right? So just compare these two cost calculations. When this one's smaller, then I'm willing to tolerate memory because it actually gives me an overall faster long-term growth rate in fluctuating environments than if I had to go through lags every time. So, is everyone with me? So in that case, you could just very generally see that if the environment fluctuates rapidly and Tn gets small, then this cost can be made small and smaller than these two numbers, which do not depend on the environment. So in a rapidly changing environment, memory is kind of generically advantageous. Yeah?
But you have more than two environments, then you will have to maintain a pool of multiple enzymes that have a cost.
Right. So now, one interesting thing is to generalize this to multiple environments, right? And take that into account. Say it again? The sewer, the gut. Right. There are many different calculations. That's right. Yeah.
What happens when the environments are more noisy? This assumes it's a frequency.
Yeah. So I'll get to that next. Absolutely. Yeah, that's an interesting point. Yeah.
What's interesting is this is all for the stability of the protein that you're calculating it on.
Right.
But another component of this is synthesis.
Right.
And the synthesis is regulated by a repressor whose half-life is very short. and whose concentration is the opposite. It's very low.
Mm-hmm.
So while this is going on at the stability level, the synthesis is a completely different set of equations,
Yeah, that's
which
true.
is relevant to this, right?
That's true. That's true.
So that the Elowitz experiment showed that you had so few repressors that when two cells divided, one could get one and one could not get one.
Mm-hmm, mm-hmm, mm-hmm.
And that there were actually variations in the population immediately
In the repressor
by
side.
the
Yeah, yeah, yeah.
repressor.
That's
So
true.
The
That's true.
rate of,
Mhm.
it's quite interesting, the rate of synthesis has evolved to be exactly the opposite of stability.
Yeah, that's very
And
interesting.
the rate
Mhm.
the rate of stability is dominant here, obviously.
Yeah,
because of
right.
the effect. But the rate of, so you're saving synthesis in some sense,
mm-hmm.
which is a cost. I mean, had you just.
That's right.
had
That's
you
right.
Had stability in synthesis, you would have increased your cost.
That's right. And so in that case, also because of the numbers, you're making so many more lac Z's, obviously, than lac I's. But I think the lac I side of it is also very interesting. So, and I think that also, actually, that plays probably a big role during this question about the rapid fluctuations how do you induce when you have very rapid fluctuations. So that's another whole side of it, yeah. Okay, so what was the question on the random environments? I think, right, okay. So this is the point about periodic, right? So you saw a purely periodic case here. Let's take a look at that in a little more detail and then look at random environments. So generally speaking, when environments fluctuate rapidly, memory we think should be useful. How much memory should the cells use? Is there some optimal value of the degradation rate? That's the basic question. How quickly should you degrade? So here's a more detailed view of what happens in this periodic environment. So what we do is we take the equations that are given here in this diagram. We take these equations and we just solve them exactly, right? That's straightforward because these are linear ODEs, so very, very easy. And solve them and then feed in the solution in one environment as the initial condition to the solution and then in the next environment and repeat the cycle. It's periodic, so this will converge to a nice limiting form and compute the long-term growth rate. Okay, so that computation can be done easily, and you can plot as a function of the degradation rate, plot the long-term growth rate in a periodic environment. Okay, so let's just take a look at that. So keep in mind, if the degradation is rapid, then I have less memory. If it's slower, then I have more memory. And this is what you see. Where are we here? Okay, I'll just press my, yeah. Okay, so what we see is this kind of curve. So here is the curve you would get as a function of degradation rate when the duration of the environments is 1. I've set 1 to be on the order of the generation time. Okay? Now I increase the duration of the environment a bit. In this case, you see that the best thing to do is to be constitutively on, so have a ton of memory. Now I increase the duration of the environments a bit, and what you see is the curve comes up and then comes up more. Still, the optimal memory is here. This is the best, this is the highest value of the growth rate. So if I were to adapt my rate constants, I would adapt to more and more memory. This is where I would want to be. But the moment I cross a threshold, you see that the curves now develop a maximum out here. And so now, for any environments that are longer than three generations, right, the best thing is to have less and less and less memory, so rapid degradation. So you see that what happened here is that the optimal strategy has jumped from constitutively on to rapid degradation with no intermediate states as I changed the environmental duration by a little bit. So that's a very sensitive behavior of the optimum. Okay, and that's the purely periodic case. So that's the prediction about the optimal degradation rate. But now, getting to the question about random environments, right? In a randomly changing environment, the durations could be short or long. So you could have sometimes the environment rapidly changing, sometimes long durations of the same stress, right? And a whole spectrum of fluctuations. And so you could imagine that there'd be periods where it's good to rapidly degrade the enzyme and periods where it's good to maintain it, right? The periods where you have rapid fluctuations, you want to have memory. So over long, long time scales, what should you do? So it turns out that in that case, there's actually going to be a finite optimum at a non-zero but non-infinite level of degradation. So you see that in this case, what we've done is we've solved this numerically in two environments where you have short and long environments, okay, and run many, many simulations and plot the long-term growth rate as a function of the degradation rate. And what you'll see is that the curve has an optimum at a certain value of the degradation rate. Okay? So in a randomly changing environment, you kind of somewhere between these two extremes. That's the basic point.
How much does this depend on the probability distribution
Of the environments. Yeah, I'll show you that right in the next slide. So what you see here is that memory is tuned to the statistics of the environmental fluctuations. We'll get to this point. The fact that actually this optimum depends on the statistics of the environment. Yeah?
How sensitive is this to choice of parameters?
It's not very sensitive. The shape, so the details are, but in a second I'll show you a phase diagram, and that phase diagram shifts a little bit, but is not sensitive. The structure is not sensitive. Yeah?
On the previous slide, could you explain again on the previous slide? The non-monotonicity when you had low DMI, the green line,
Yeah.
for example, in DMI of 1, while you get much worse off, you degrade a little bit more and then you start getting better once again.
Yeah, so that starts getting into the fact that you now have a mixed population of cells that could be actually like in the wrong phase with the environmental fluctuation. So basically you're now maintaining a diverse population, and so there can be selection that pushes up certain states in the environment, but then they become dominant in the next environment. Yeah.
Go out of that.
Yeah, that's exactly right. Yeah. Okay, so let's talk a little bit about fluctuating environments. And now, just one slide to kind of give you a sense for the calculation. Because the periodic case, I could just explain to you right in words, and it's clear that you could do it. You're just solving an ODE in two environments. That's easy. But in a random fluctuation, these durations are random variables. Durations tau are random variables with some distribution. And so what happens is, you can go from here to here, and then go to different initial condition, different final condition, and you see that this trajectory is a complicated thing, right? And it's going to depend on the statistics. So this is a problem of how to compute the long-term growth rate. If you know the statistics of this trajectory, you could of course do it, because at every point here, you know how quickly the cells are growing. So that's doable. But what you need to know is a way to capture these statistics of this trajectory. So exact calculation is not possible in general. But what you can do is you can develop approximations that are analytical and allow you to then develop a phase diagram. So that's, I want to give you a sense for one way to do this. So here's what you can do. You can solve the ODE in any environment, E, subject to any initial condition, okay, for a certain duration. So this is something you know the solution, right? This is very, very simple linear equations. So you know the solution in either of these environments depending on this initial starting point. So now you need to do some kind of averaging because the environment is a random variable. So what you can do is you can say, suppose I start with a specific initial condition, which I'll fix for the moment, okay, solve my equation perfectly for time tau, which is the duration of the environment. So solve it for this time, starting from specific x, right? And then you end up somewhere, which is given by this, right? Now, average your endpoint, okay? So take the average over the duration of this interval. So you now have an average value for this endpoint. So what we have here is the average value of the endpoint conditioned on having started at a specific x0, okay, in one environment. And then the same thing for the other environment, right? Conditioned on starting at a specific point in the next environment, right? Figure out the average location where you end up after that environment. Okay, so all that is in this integral here, right? Now the approximation is to say that I want to end up on average where I start, okay? And that's a self-consistent set of equations, okay? So say that your starting point should be in the next environment should be the average endpoint of the previous environment in both directions, okay? Solve those equations and then see how well you do. So this is how well you do. So basically the dots here are the numerics, and the gray curve is the analytical, this approximation, which we call mean field. All right? It's mean field because you're treating your initial condition in each environment as kind of an average. All right? So it does pretty well, except in some area around here, and you can improve this approximation by taking into account two environments. All right? So you can say, I want to treat the previous environment as well. Right? And so you could eventually iterate that. So actually with two of those, just going back once, you're already quite good. So the dotted line, right. Okay, so we use this basically to now make a phase diagram, because if you have an analytical expression, it's easy. You don't have to scan parameter space, and you can just plot things. And so what we do is we say, how does the response strategy depend on the environment? So we'd say, okay, this is getting to the question on statistics, take a gamma distributed duration, all right? So the gamma distribution has two parameters which control the mean and the variance. So it has many, many different shapes are possible, exponential, right, or bump, or almost normal. So all these shapes are possible, and you're controlling with two parameters, the variance and the mean. So this is the mean duration of the environment. This is the variability of the durations. And every point here is a different kind of randomly fluctuating environment. And we'd like to know is which strategy is best. So by using that analytical expression, you can find this kind of phase diagram. And you can see that it's best to be constitutive in this portion and memoryless here. So keep in mind, if I go to 0 variability, I am a periodic environment. So this axis is the purely periodic case that I showed you initially. And you see that we have this transition from being constitutively on to memoryless at a critical value of the environmental duration. Now, what happens is, so there's only two phases here, right? Now, as I start making the durations more variable, so I have more variability in the environment, right? This phase is still stable, even to some noise in the environment. You still want to do constitutive. But at some point, if you have too much noise in the environment, you have a new phase, which is response with memory, with that finite amount of memory. And this is, like any phase diagram, you have different transitions. So I already showed you that this was first order, discontinuous jump. And turns out that here you have a continuous transition of the parameter. Here you again have a first order transition if you go from these phases. And then here you actually have a continuous transition. So the character of this transition actually changes. So you actually, this is kind of your phase diagram, first order, second order, first order, second order, and a point where things change. And so biologically, I think this has, these transitions are interesting because they mean that, an abrupt transition means that you lose, you change the whole way that the gene is regulated, right? Kind of a larger transition. A continuous transition means you're adapting the parameters of the system. So that's kind of just a kind of hand-wavy way of thinking about it. Okay, so that's basically the summary, is that sensing with memory is a strategy that's optimal exclusively when environments change randomly. So that's actually one upshot of this, is that only when you have enough randomness is it useful to have some memory. Okay, if I don't have randomness, if I have still variation in the environment, durations that are periodic, right, then finite amount of memory is not good in that case. You really need some amount of unpredictability in the environment in order for memory to be useful. Yeah?
How does the maximum growth rate evolve in the diagram? Like which phase would correspond to best conditions for growth?
Oh, that's a good question. So I don't have that actually plotted here, but that's something that can be done. I don't have that information, yeah. But this did show you the, yeah, so the only extra piece of information here is that these are the lines of constant memory, okay? But does not answer your question. Okay. And the other point here is that the changes in the environmental statistics, so in other words, how, just the statistics of the environment, not even the environments, just the way that the environment changes, that can drive the evolution of the network in different ways. And the third point, which really has to do with the fact that these jumps can occur at such a narrow range of changes, right? So you can go from an environment with a mean of 2 generations to 3 generations, and suddenly the best strategy completely changes in this simple model, right? So that indicates that there is some kind of sensitivity to the system. Okay, so I left time for a few questions, but there were a lot, so we can also go on. Yeah?
So how would something like circadian clocks fit into this framework? Because that's
Right.
presumably like better than circadian.
Right. That's really anticipatory for periodic environments. So, yeah, if you had, you could tune networks to do better than this, is the point, right? So this is the simplest. You saw the model has 2 rate constants. So you can, of course, make much more refined networks that'll probably beat any of these,
Yeah.
right? So, and we see such things, right? And real networks are much more sophisticated. So the question is, are there some, adding, competitions with circadian clocks, you could estimate how long do you have to wait until circadian clocks would take over. Kind of like, what's the advantage? In some cases, you could probably see very quickly that's a very big advantage. Yeah. Okay. So the second part will be on stochastic switches, and
There have been suggestions of clocks in bacteria, right?
like the cyano clock. Yep.
Yeah. So I mean, it's kind of interesting that could have been added in if they had the machinery. Maybe I don't know if E. coli has any machinery that looks like a clock, but
I don't know of any.
an
Yeah.
oscillator
Yeah, you just need an oscillator, right? Well, the E. coli has one oscillator that at least that I know of, which is this the min system that determines the midpoint. But that's
it's interesting
yeah,
that you could think of ways that this circuit could get more sophisticated.
yeah, absolutely, yeah,
I guess it's again cost benefit.
yeah, yeah, yeah. Because the circadian clock, has its costs, right? You have to. Yeah, so there's a whole field of cyanobacteria, how the clock is regulated, and the costs. Yep. Okay. So the second part will be 15 minutes left, so I'm going to just show you some of the experiments, tell you a little bit about the basic motivation for looking at the next set of experiments. So basically, as I mentioned in the beginning, if you have stochasticity, right? So we talked about responses and responses with memory. If you have stochasticity where the cells can randomly change their state, and you have a big population, that's actually quite useful. So, and in any case, and I think we got to it in one of the questions, if you already make some cells which are in the wrong state and you maintain them for some period of time, right? They're going to influence your population growth rate. So, in all these cases, you have an interplay of the response and selection. And by selection I mean the fact that some cells are growing faster than others and their states are heritable for some time. And that's all. So if you have any amount of heritability plus the fact that there's growth rate differences, then you have selection. And on top of it, you have physiology, which is the fact that you can change the states and all that. And so the question is, the population adapts with both of these mechanisms. So you could ask which one wins, but you could also ask, what happens when there's mixtures. So usually in evolutionary theory, these are considered really separately. So physiology is thought to act fast. It determines the phenotype of the organism over the lifetime. And then selection acts slower time scale between the generations. And in evolutionary theory, there's always the attempt to give a single number that summarizes this. This is the fitness effect. So if you have a mutation which changes the physiology, you figure out how it acts on the phenotype and try to summarize that with a single number which tells you how quickly will that mutation take over, or how quickly will that mutation get lost. So that's called the selective coefficient. And I think you heard about that in the previous talk. So in reality, you know that there could be complex interactions between the physiology and the selection. And these are not often studied. So we'd like to know what are the relevant timescales that impact these interactions. And practically speaking, how much detailed information do you need in order to predict the outcome of the evolution? So there's a nice way to kind of mimic a lot of this on rapid timescales. The problem with doing the experiments that we really want to do is that they would take very, very, very long, because mutations in bacteria, the mutation rates are very low, 10 to the minus 10 per base pair, right? So 10 to the minus 3 per genome replication, right, which is extremely low per generation. So you have to wait a very long time for mutations to come in. The mutational effects could be small, right? You won't be seeing things for a long time. And then you have billions and billions and billions of cells. So how will you know what happened? Okay, so there are different ways to deal with all that, but there's a separate direction, which is you build a system in the lab that has a certain amount of heritability to it, okay, and it also has certain costs and benefits, and you can track the cells. So that's the kind of direction I'm going to show you here. So you make a synthetic system, and what we did is we took this very nice system that was built by Han Lim and Van Oudenarden and published in 2007, where they studied a stochastic bistably expressed gene in E. coli. So this is a gene called AGN43, whose promoter has been studied quite a lot, and the promoter can get methylated, and when the promoter is methylated, the gene, whatever's sitting here, is on. When the promoter isn't methylated, then a gene called OxyR can bind there and repress this gene. Okay, so OxyR protein can bind the promoter and repress expression of the gene that's sitting here. So when this OxyR protein is bound here, it actually occludes the methylation site. So it makes it very difficult for this gene to get methylated, and you have to wait until this falls off, okay, as an off rate. So it falls off at some point. And then you have a protein called DAM that methylates the sites that are sitting here. And now it's a competition between, does OxyR bind back, or does DAM methylate it? If DAM methylated it, then it's going to get methylated and prevent OxyR from binding. And if OxyR got there, then it's going to repress it. So you see that these states, and methylation is heritable, right? So basically when the cells divide, this gene will become hemimethylated, and then DAM will rapidly methylate it, right? So it kind of has to miss 2 methylation cycles in order to allow OxyR to rebind. You see, you have this kind of bistable state. You can either be on or off, bottom line, okay? Bottom line is you can either be on or off. And so the gene that normally lives here is called AG43, and it's a cell surface protein that drives cell-cell aggregation. It's a very interesting gene. But to make a synthetic system, they actually knocked out that interesting gene and replaced it with the T7 RNA polymerase, which is a polymerase from a phage, okay? And so that's from a bacteriophage. So this polymerase now gets turned on stochastically, and when that polymerase is on, it'll turn on T7 promoters. So now what they did is they gave a plasmid, they supplied a plasmid into this bacteria that has a T7 promoter that drives GFP, so they could visualize and see when this gene is on or off. This is actually an amplifier, because T7 RNA polymerase is very, very powerful polymerase, makes a lot of protein. And so if you give a T7 promoter, you have a ton of GFP in the system. So what we did is we took that. This is a very, very nice paper that studied the switching of this locus. And what we did is simply added one more plasmid. We added a plasmid that gives you tetracycline resistance under T7 control. So what that means is when this turns on, it's also resistant to the antibiotic tetracycline. And you can watch the cells. So here's a little movie showing you a single cell sitting in a mother machine, and you see it's dividing, and now it turned on. And now it's going to be heritable. So you see many cell divisions where it remains in the on state. And you can just count how long before it'll become completely red again. And if you count, it's about 20 or so cell divisions, and it just turned red. And now the cell is heritably red. All right, so you've just watched a promoter change from on to off in a single cell. And if you do the statistics on this, you'll see that that was a very rapid switch there. It typically takes about 100 cell divisions before this switches one way or the other. And there's some bias between the two rates. Okay, so now what we did is, since we added tetracycline resistance, every time the cells are green, they're actually resistant to the antibiotic. So now we put these into our bigger chambers, and we give pulses of antibiotics. And in this case, rapid pulses. So every 6 minutes, we give a pulse that lasts for 6 minutes of the antibiotic with a certain concentration. This concentration is not hugely high, but it's above the minimal inhibitory concentration, which is where biologists typically define as the antibiotic is acting strongly. All right? So if you're above the minimal inhibitory concentration, the cells will not be able to grow. They will not be able to grow as a population. In single cells, you'll see that they actually do something. So what you'll see is you're getting rapid fluctuations here, 6 minutes, kind of square waves. And here's what happens. You're getting a lot of tetracycline, so the cells are actually filamenting. You see they're going out. You're not seeing many cell divisions at all. They're just filamenting, and they're making funny shapes. Okay, so making funny shapes. Some of them are lysing. They can all turn on antibiotic resistance, and when they do, they'll be green. And once they turn on, you see a selective sweep, where that population is now dominated by green because it turned on the resistance genes. These populations go extinct because they randomly didn't turn it on in time. Here's another population that does a sweep. Okay, and other ones go extinct. And you're also seeing here counter sweep, where cells that are switched back actually doing pretty well. And you already saw in the beginning of the movie that cells that are actually off are actually doing not too badly for 10 hours, right? For 10 hours, they did pretty well. So there's a lot of dynamics here in a very, very simple system where you have heritability. This is, just to give you the time scale, this is about 30 hours. So you can keep going, and you can actually study this now in many, many populations. So this is just in high resolution, but you can now go a slightly lower resolution and study 80 populations at the same time. Okay, so then you can get statistics on the dynamics. So for example, here are a few different populations, and we're just tracking them with 3-hour fluctuations. So you see stress, growth, stress, and you see this one develops some this is the on-cell frequency, how many are green. So this one got a lot of green cells initially. These ones didn't, and eventually they get a little bit, and eventually they do. You see, so sweeps of the on state can occur at different stages. And I'm calling them sweeps because they're heritable. They're really like a genetic sweep. It's epigenetic sweep. Okay? And then we can also track the growth rates of the cells. And you see that once they get on, they basically become immune to fluctuations. The blue curve here doesn't oscillate with the environment, whereas the other ones oscillate until they develop sweeps. So that's basically the setup. And then you can start asking, when do you see sweeps more frequently? So you saw the condition of 6-minute fluctuations right here, and this is telling you how many on cells were in the first 12 hours, so very few, more and more. So as you saw in that movie, eventually you got more and more on cells in different populations. So this is telling you how many populations have on cells during the experiment. So that's for 6 minutes, 12 minutes. But then once you get to intermediate periods, you see very few sweeps occurring until you get to longer durations. And even then, you don't get as many sweeps. So this was very surprising to us, that basically, you'd think that if you had a lot of time, then you'd start getting a lot of sweeps. So that was surprising. We also found that the cells, the long-term growth rates are actually largest at these intermediate periods. So if I'm fluctuating, and this is, by the way, on the order of the generation time, 30 minutes in this experiment. So you see that that's where the growth rates, the long-term growth rates, are actually largest. So that was surprising to us. We didn't quite understand that. And I should say, and I think this is another place where there's a lot of room for, physicists and mathematicians to understand what's going on here. We really can't explain any of these dependencies with simple models. So this is kind of the antithesis of the previous part of the talk where we had a very simple model. But if you want to try to measure things in experiments, you suddenly realize, wait, the dynamics are not quite right, and I can't even predict things like the peak of the growth rates at intermediate periods. How frequently do I get sweeps, right? Clearly, it seems we need to know more. So what we found is in this experiment, you need to know quite a lot more because the off cells, the on cells are simple. Once they're on, they'd grow in all the environments. Very good. But the off cells, you saw that the cells that didn't have the green, they already could survive for 10 hours, which is nice. These cells are just E. coli cells with no resistance to tetracycline. And for 10 hours, they're happily, not so happily, but they're surviving. They're making it, right? So if you fluctuate just on susceptible cells, you see that actually they go through some interesting dynamics in single cell level. So now we tracked all the cells, and we're telling you how quickly they elongate as single cells. And you see when they get stressed, they turn off the cell division rate to 0 immediately, but they still elongate, not as rapidly, but they still elongate. So what that means is the cell size grows. Then when you switch them back to growth mode, they resume divisions after some delay, and then their cell size goes back down. Okay, so single cells eventually get into this kind of oscillatory dynamic here of elongation and then division, and it starts being in sync with the environment. That's what happens when you have periods of around 3 hours, which is about 6 cell divisions in growth here. If you go to rapid fluctuations, like I showed you before, what you see is filamentation, which you saw in the movie. Cell divisions go to 0, but elongation, while declining, does not reach 0, and therefore cell size grows indefinitely and eventually filaments. Okay? And these filaments are much harder to recover than things that are just a little bit larger than normal. So at some point, there's other work that has shown that there's some kind of transitions also with filaments, and filaments can become kind of crossing some catastrophic threshold and things like that. So we haven't studied that properly in this experiment, but that's certainly already known. So there's basically these two kind of modes. One is kind of you go into filaments, the other is you can recover and keep your cell size in a decent range. So that process actually determines a lot about the evolutionary dynamics because, right, you can see that now you have a changing growth rate within single cells, and that's not captured by the model. The model was a two-state model, right? The question is, do you need to put this in? Is this going to allow you to predict the long-term growth rate?
The first is very interesting. The first case is the effective growth rate of a single cell is much smaller. It still does this pause. So if stochastically you generate a green clone, it will divide much faster, right?
Yeah, absolutely.
So won't it have an effective fitness advantage?
It will grow much faster than this, right? But it will not grow much faster than this.
Sure.
So, and the question is, but while at the same time, there's a cost to that tetracycline resistance, which we later measured to be around 10%.
Oh, I see. That's
So,
in unstressed conditions.
yeah, in the unstressed conditions. So, and that's really because of what I said about the T7 RNA polymerase, which just makes a ton of this protein. So we kind of didn't build it in, but it came out that there's, in this system, a cost, which is nice because we like to think of cost and trade-offs. But you can actually make, you can improve the TET system, use a more like the wild-type TET, and then you'll find that, indeed, the cost is not at all as big. So just to show you briefly before wrapping up how you can imagine, you want to go beyond the simple models, what can you do? So in this case, from these experiments, you see that you have a dynamic elongation rate. And so we want to see, can we write down an ODE model for the elongation rate? That's kind of the first thing you could imagine solving. What are the dynamics of this changing variable? And so you can, since you have single cell data, you can actually plot elongation rate of single cells versus the derivative of the elongation rate. in the two different conditions. So this is the result from single cell measurements. So you kind of get the actual plot of how this looks. So you notice that in the growth phases, the recovery in growth media, you see that slow growing cells, they recover, means they change their growth rate slowly, and therefore the growth rate will grow, and it'll grow until it reaches here. The derivative here is 0, and you see it's a stable fixed point. If you go above it, it's going to reduce, and if you go below it, it's going to increase. So that's a stable fixed point. If you have killing phase, which is you give antibiotic, then slow growing cells kill slowly, and fast growing cells kill fast. So the bottom line is that the more stress the cell, the slower it recovers and the slower it gets killed. So basically stress cells do everything slower. And that's good because it's good to not get killed quickly. Okay, so you see that having some cells that are stressed can be useful in fluctuating environment, and that actually allows you to understand why they can actually adapt, right? So the off population in this experiment actually has this adaptation kinetics and can survive for some periods because it maintains some slow-growing cells. Slow-growing cells give them an advantage. Okay, so the bottom line is we tried to predict the total. This is really just summary of all the different periods, showing the growth rate plotted on some unit period. So this is measurements over many populations here, and then trying to fit this kind of simple quadratic model that can capture both of these, just fit that to all the data. So again, there's 6 parameters here, so it's already quite complex, but it fits decently well, but not fantastically. And you do pull out, just from the total bulk kinetics, you pull out these 2 dependencies which would reflect that single cell data. So this came just from bulk population growth rate, but it gives you back the ODEs that you can actually see in the single cells, just qualitatively, pretty nicely. So now when you take everything together, you throw everything I just showed you together to predict the long-term growth rate over the entire experiment as a function of the period. So the data are the points with error bars as the variance between the populations. And the ODE prediction is this guy. And you see that, in fact, the ODE is in fact this guy here, so it doesn't do very well at all. And the only way you get these rapid periods here, so in other words, the very rapid fluctuations, the only way you get that correctly is if you say that when the environment switches, the internal state of the cell doesn't feel it immediately. And that makes sense, right? Because if the external environment switches, from tetracycline to growth media without tetracycline, then the cells still have a ton of tetracycline inside. So there's still internal kinetics of getting rid of that tetracycline that we find has a time scale of around 1 minute from fitting. And only if you put that in do you actually get this side of the curve. And you still miss this very strong effect at intermediate periods. So that's the show, and it's kind of just to show that there's plenty of open room to understand really what's happening at the interface of adaptation and physiology here. And that we don't understand, for example, this very pronounced peak. And, again, we're putting in kind of a few parameters and trying to fit things phenomenologically, but there's of course another layer here, which is the actual mechanism, how do you become resistant and all that. Okay, so let me just wrap up by saying that in this case I showed that we build a simple stochastic system, simple synthetic switch. It gives you a practical model that you can actually study in the lab. It has all the features, some of the features of real evolutionary dynamics, where you have heritability, diversity, growth rate differences, rates of switching, all that stuff that you see in evolution, but you can track all the cells. And we do see this very high sensitivity to the environment in this experiment, right? So basically we change from 6 minutes to 15-minute fluctuations, and immediately you don't get sweeps. So that's something very sensitive, and you see it already in this simple case. And the last kind of big point is that the physiology in this case has a really pronounced effect on selection. So the fact that the cells could adapt on their own without the mutation, but it depends on how the environments look. That's really the fact that physiology tells you a lot, really has a big input into the fitness, and you can't, in this case, represent it with a single number, right? You have to, even if you write down a full ODE, you still don't quite get it. So that kind of opens big questions for how do you really do evolutionary dynamics on time scales that are, intermediate. And okay, that's the reference. And this work was done by Weishan Lin, who was a PhD in the lab a few years ago. And everything else I told you, the theory was done by Antun, who's here, and Guillaume did the experiments on the lac fluctuations, and we got some help from Colin Jouet, Toby Bergmiller in IST Austria, and other collaborators for other projects that I didn't get to today. And thanks a lot. Yeah.